All right, everybody, welcome back. We're about to start part 2 of the hematology section. And in this section, where the majority of the time is going to be spent talking about hemostasis and the clotting cascade. But before we jump into that, my dog Coco wanted to say hi. And in fact, she does not like sitting in front of the laptop anyway. So she thinks she's always in trouble when I do this. But anyway, here's a Coco sighting. And I'm going to hand Coco off to my daughter. Thank you, Sam. Alright, now let's jump into this section. This part 2. So we're going to start off looking at leukopoiesis, which is basically just the formation of leukocytes or white blood cells from the stem cells in the bone marrow. And granulocytes, as we saw a little while ago, they'll mature in the bone marrow and then get released into the blood supply. And then finally, agranulocytes are released into the bloodstream before they fully mature. So think about like the T and B cells. They are released by the bone marrow before they're fully active and fully mature and so on. So in looking at the lineage line again, just review that the common lymphoid stem cell will give rise to lymphocytes. Common myeloid stem cell will give rise to all of the other blood cells. And in the last section before we transition totally into hemostasis and the coagulation cascade, let's look at the formation of platelets or thrombocytes. So thrombopoiesis. Do you remember where thrombopoietin is made? And if you're thinking the liver, you're right. The liver makes most of the thrombopoietin, and then that hormone will stimulate bone marrow to make more of these particular cells that we call megakaryocytes. And it's the megakaryocytes that will actually create all of the thrombocytes or the platelets that we know. So what a megakaryocyte is, it's a very large blood cell that's still found in the bone marrow. And what will happen as the megakaryocyte matures, it will start piecing off fragments of itself. And those fragments are what we call thrombocytes or platelets. So just going back one slide to this chart, if we look at where the megakaryocyte is right here, this doesn't really have it drawn to scale. Megakaryocytes are much larger than red blood cells, mast cells, and all of these other blood cells. And in the formation of platelets, when they're making platelets, they will start piecing off cytoplasmic fragments of themselves. And inside of those cytoplasmic fragments are a bunch of goodies. So let's look at these platelets here in more detail. Oh, I forgot to mention this term, endomitosis. That's the process of a megakaryocyte breaking off fragments and pieces of itself to create these platelets that we know of. So here's an image of platelets. This one is round. There are no extensions coming off of this platelet, so this platelet is not activated. However, this platelet over here on the right definitely is activated because we can see it's sending out these protrusions, and that's one of the characteristics of an activated platelet. So I want you to think of platelets as goodie bags. They're just little cytoplasmic fragments, but inside of these platelets are all kinds of goodies. And these goodies help with clotting, and they help with tissue healing and repair, and they help with a number of other things. So keep that in mind as we go along. So as far as platelets go, remember they're stimulated by thrombopoietin, which is made by the liver, and then platelets are really only good for about 10 days after they fragment off of the megakaryocyte. So we're always making platelets all the time, all day, every day, making platelets and red blood cells and white blood cells. So now let's look at the big topic of this section, hemostasis. And the word hemostasis basically means to stop bleeding. And this is a really good thing. If we injure a blood vessel or we injure a tissue and blood is now leaking out of that blood vessel, we need that leakage to stop. And the process that the body uses to stop that bleeding is called hemostasis. And there are several requirements that are needed to have effective hemostasis. Number one, we need platelets, and we need enough platelets, and we need platelets that can become activated. And then we also need clotting factors, and these are proteins that are pre-made that participate in the formation of a clot. We also need adequate blood flow to the area to get platelets and clotting factors to the area that's bleeding. And shear forces, the forces that may have injured that blood vessel to begin with, also help to initiate and start the hemostasis pathway and the coagulation pathway. So we'll look at that process as well coming up. And obviously we need endothelial cells because damage or injury to endothelial cells will initiate the formation of a blood clot and hemostasis. And then finally, we need a way for the blood clot or that platelet plug that forms to be broken down. And so we call that process fibrinolysis. Okay, so starting general, and then we'll get into specifics. So here are the basic 3 steps of hemostasis. Number 1, when there's a damaged blood vessel or a torn blood vessel or capillary or whatever, we have vascular spasming happening or vasoconstriction occurring right away. And this is only a temporary vasospasming or vasoconstriction. So maybe you've recognized this on yourself or on a friend or a loved one. Think about a situation where maybe you've fallen down and scraped your knee or elbow, and you look at the area that got scraped immediately after the injury takes place. It usually doesn't start bleeding right away. So if you scrape your elbow and then immediately look at it, what you're most likely to see is, the damaged skin and tissue, but it's going to be pale. It's not going to be bleeding yet. And that's because at the very beginning of a traumatic injury like that, vasospasming occurs, and that limits the amount of blood loss initially. But as you keep looking at that injury, then you will start seeing bleeding kicking in, and it starts to hurt and so on. So first step, vasospasming, vasoconstriction, and that kind of reduces the amount of blood loss. And then the second step is the platelet plug formation. And this occurs when platelets become activated and they form a plug that will basically plug up the hole that's in that vessel or plug up the end of that capillary that just got torn away and whatnot. So that helps to definitely limit the amount of blood loss as well from that injury. And then the third step here, which actually kind of starts at the same time as platelet plug formation, is the formation of a blood clot or a fibrin clot. And this occurs because of the coagulation cascade or the clotting cascade. So we're going to look at the specifics in mostly step 2 and 3 here, and I'm going to highlight the things that I really want you to know about these things. So once we have a platelet plug formed and a blood clot forming, that's when the clot and those platelets and other white blood cells can initiate repair to the damaged area. So let's look at this example right here. We've got a cutaway view of a blood vessel, and we have a torn tunica intima layer, torn endothelial layer, and this tissue damage is what's going to stimulate, number one, vasoconstriction. So these smooth muscles around here in the tunica media of this vessel are going to vasoconstrict, and that'll limit the amount of blood loss potentially from this injury. And then secondly, we've got platelet plug formation, where I'm going to go back one slide. We can see these platelets right here are not activated. They don't have any extensions coming out of them. But once the platelets do become activated, they send out these arms and they become sticky with one another. and they start grabbing onto each other and grabbing onto red blood cells and grabbing onto endothelial cells. And as they continue to grab on and stick to one another, they form what we call this platelet plug. This always reminds me of the lazy river. I don't know if you guys have ever been in a lazy river, but a couple summers ago, I was in the lazy river with my family. We were at the water park, and there's always those kids in the lazy river that grab on to their friends, and then they grab onto the sidewall of the lazy river, and they form this mass or this obstruction. And this happened last time I was in a lazy river, and initially I got frustrated because they were blocking everybody from moving forward in the lazy river, but then I realized that this is a platelet plug formation. They were demonstrating that. So that's exactly what's going on when platelets become activated. They start grabbing on to their friends and creating this plug, or this clot, if you will, that traps other cells and keeps them from moving along. And what it ends up doing is creating this plug that's going to block the loss of blood from that damaged vessel. So then the third step, remember, is the formation of a fibrin clot. And this is made possible by way of the clotting cascade. So these string-like structures that you see in this image, these are fibrin strands. And fibrin is the end product of the clotting cascade. And what the fibrin strands do is stabilize this platelet plug, which we now call a clot, and keep it in one place and make it strong. And once it's secured by these fibrin strands, this meshwork or this netting of fibrin strands, then the platelets can start releasing their healing factors and so on, which will help to initiate the repair of the damaged vessel and damaged tissue there. Okay, so in word form, let's look at this. So number one step, vasospasming or vasoconstriction, that limits the amount of blood loss from that damaged area. And then platelet plug formation, that's the second step. And we can break this step down into its various components. So initially, platelets need to be activated. And one of the things that activates platelets is calcium. And if we've got damaged tissue and broken cells, we have cells that are releasing calcium. And it's that increase in concentration of calcium at that damaged area that will initially activate platelets and make them start grabbing onto one another and grabbing onto other things, making them sticky. And then we also have adhesion made possible by von Willebrand factor. And if you're familiar with von Willebrand factor, you might remember that von Willebrand factor is made by endothelial cells, and when endothelial cells get damaged or destroyed, they'll release von Willebrand factor, and this is another component that really enhances the stickiness of platelets. So this allows platelets to stick onto the endothelial cells around where that injury or where that damage took place. And so that's what will help keep the platelet plug in place while the fibrin clot is forming. So we need von Willebrand factor to enhance the stickiness of platelets. And so then it goes through another activation stage, and then they aggregate, sticking onto one another, kids in the lazy river grabbing onto each other. That's what's going on here. And then finally, the last stage of platelet plug formation is the secretion of granules. And granules meaning that that's when the goodie bags, which platelets are, they open up and they release all of their goodies. And those goodies are, they can be neurotransmitters that can reduce the amount of pain that's felt in the area. They can be healing factors. They can also be clotting proteins that help enhance the formation of a blood clot. So that's why I consider platelets goodie bags. Initially, they'll stick to one another and make a platelet plug, but then when they degranulate, they're releasing their goodies, and all of those contents help to stabilize that clot and to promote healing and repair. All right, I know this is a bad copy. You don't really need to read these words, but you can try if you want. Let's look at the pictures though. So over here we've got damaged blood vessel, endothelial cell damage, and calcium release, and von Willebrand factor release, and that's all going to activate platelets that are normally in the blood to become sticky and start attaching themselves to this damaged area, creating that platelet plug. And then at the same time as these platelets are becoming active and sticky with one another, we have the development of these fibrin strands as a result of activation of the clotting cascade. So as the platelet plug is forming, we also have fibrin strands, or this fibrin netting or meshwork forming that stabilizes this platelet plug, and then we have what we call a blood clot, or a full-on clot, where the fibrin strands are keeping that platelet plug in place. Platelets then will start to degranulate, releasing healing factors and repair factors and proteins that stimulate healing and repair, and so on. So here's a list of all of the goodies that are found inside platelets, and I'm not going to ask you to memorize this stuff. Just take a look at this. Inside platelets, we've got fibrinogen, which we'll look at in just a second. That's the precursor to fibrin. Platelet-derived growth factor. This is a protein that stimulates growth of new cells, so it aids in the repair of that damaged tissue. We've got ATP in there. We've got histamine, serotonin, epinephrine, all kinds of goodies that support the healing and repair mechanisms so that we can have rapid resolution of that damaged tissue. Okay, so now let's look at the clotting cascade, the third step in hemostasis. So I've got on here four most important players that make up the clotting cascade. Let's look at these. So thrombinogen and thrombin, and fibrinogen and fibrin. Sometimes you might see thrombinogen known as prothrombin. Those are two names for the same thing. So when you see the suffix ogen or inogen, what are you thinking of? Hopefully you're thinking that this is an inactive protein or a precursor protein, which is exactly what this is. So thrombinogen is the inactive form of thrombin, and then fibrinogen is the inactive form of fibrin. So the clotting cascade is a sequence of events that will activate thrombinogen into thrombin, which will then convert fibrinogen into fibrin. And fibrin is what those strands are made up of in the previous slides for the blood clot formation. So these strands are made up of fibrin. So fibrin's the end point of, or the end product of the clotting cascade. So let's look at this clotting cascade in more detail. There's two pathways that are involved here, the extrinsic pathway and the intrinsic pathway. So you do need to know the difference between these two pathways and what makes them different. And when we're looking at a cascade of events, what that's really describing is that something will initiate or activate an initial protein, or in this case a clotting factor, and then that active factor will then go on to activate another factor, which will activate another one, which will activate another one, until finally, when we're talking about the clotting cascade, we get activation of fibrinogen into fibrin, and then it's that fibrin that's making up the strands of that meshwork, or that fibrin netting, that's stabilizing the platelet plug and what we call the blood clot at that point. So let's look at the two pathways. These are the two ways that the clotting cascade gets started. So the extrinsic pathway starts when there's damage to endothelial cells. There's a lot of things that can damage and irritate endothelial cells. So I'll let you guys work on your own to kind of review what can damage endothelial cells. But there are a number of things that we've gone over already in class. So if we've got endothelial cells that are damaged, they're not dead, but they're damaged, they're hurt, they will release what's called tissue factor. And then tissue factor will activate clotting proteins that are already found circulating in the bloodstream. And looking at the intrinsic pathway, this starts when we've got exposure of subendothelial collagen primarily. So we've got vascular injury here, not just damage to endothelial cells, but full-on vascular injury. So when you fall off your rollerblades and skin your knee and you've torn away part of your skin and torn away part of the capillaries that are associated with the skin, that would expose subendothelial collagen. The collagen that endothelial cells rest on normally are now exposed, and that will activate another clotting factor called factor 12 when it comes in contact with that subendothelial collagen. All right, so on the whiteboard, what I'm going to do is draw out the intrinsic pathway of clotting cascade and the extrinsic pathway for the clotting cascade. And remember that it's a cascade of events, and so we've got these proteins, these clotting proteins that are already made by the liver that are floating around in the bloodstream that just aren't activated yet. And so we need something to activate these clotting proteins so that we can form a clot. So if you remember from the previous slide, the intrinsic pathway gets started when we've got subendothelial collagen exposure. And that really only happens when there's significant trauma or damage to the blood vessel. So when we have exposure of subendothelial collagen, then what that will do is we already have preformed factor 12, but once factor 12 comes in contact with that subendothelial collagen, it will activate into factor 12A. So the A just simply means that it's the active form of this clotting protein. And then factor 12A will go on to activate several other clotting proteins that I'm not going to really go into detail on, but ultimately what factor 12A will initiate is the activation of factor 10 to factor 10A. So factor 12 gets activated when it comes in contact with subendothelial collagen, and then eventually activated factor 12 will lead to the events that will cause the activation of factor 10 to factor 10A. And then once we have factor 10A activated, then we have prothrombin getting converted into thrombin, and then thrombin will take fibrinogen and convert that into fibrin. And fibrin is the end product of the clotting cascade. This is what the fibrin strands are made of when we're forming a blood clot. So it's the fibrin strands that are forming that meshwork that stabilizes the platelet plug and forms the blood clot. So that's the intrinsic pathway. Subendothelial collagen exposure activates factor 12 into 12A, and then eventually that will activate factor 10 into 10A, which will take prothrombin and convert prothrombin into thrombin, and then thrombin will convert fibrinogen into fibrin, which is our end product for the clotting cascade. So let's look at the extrinsic pathway next. So extrinsic pathway has to deal with endothelial cell damage. Damage, irritation to those endothelial cells. And what damaged endothelial cells will do is they'll release a substance called tissue factor. And tissue factor will interact with factor 7 and convert factor 7 into factor 7A. And then factor 7A will interact with factor 10 to create factor 10A. So what I want you guys to know and to remember is what activates extrinsic and intrinsic pathway, and also the factors that are involved that I have written up here on the whiteboard. So tissue factor is released by damaged endothelial cells, and when tissue factor interacts with factor 7, it becomes active factor 7 or factor 7A, which will then activate factor 10 into 10A, and then once we have factor 10A, we've got the conversion of prothrombin to thrombin, and then thrombin will convert fibrinogen into fibrin. So also, while you're studying these things, keep in mind that all of these clotting factors in their inactive form are being produced by the liver all the time. So we have those floating around in our bloodstreams all the time and just waiting to become activated. So we need these in inactive form so that we're not forming clots at the wrong time. But also we need them in an active form so that if we do need to form a clot, we have these factors ready to go pretty much immediately after intrinsic or extrinsic pathways are initiated. All right, here we are back to review what's going on with the clotting cascade. Again, remember, there's more steps here than what I want you to remember. So just keep it general. Know the intrinsic pathway, know the extrinsic pathway. Know what's stimulating or what's causing the activation of these two pathways. So as far as the intrinsic pathway goes, this occurs when there's exposure of subendothelial collagen. And so vascular damage for sure, vascular trauma. When factor 12 comes across exposed subendothelial collagen, it will activate. So this is factor 12 is inactive, but is always made by the liver so that we have some there ready to activate in case we've got a damaged blood vessel. So anyway, factor 12 will come across subendothelial collagen and it will activate into factor 12A, which will then activate other clotting proteins, ultimately which will activate factor 10 into factor 10A. And once we have active factor 10, then that will convert prothrombin or thrombinogen into thrombin, which is the active form of prothrombin or thrombinogen. And then thrombin will then take fibrinogen and make fibrin, or convert fibrinogen into the active form of fibrin. And then we've got the formation of these fibrin strands. So intrinsic pathway starts when factor 12 comes in contact with subendothelial collagen. Extrinsic pathway, on the other hand, is initiated by endothelial cell damage. So endothelial cells that maybe aren't dead or destroyed, but they're damaged, they're irritated. And so then they will release what's called tissue factor, and tissue factor will basically activate factor 7 into active factor 7, factor 7A, and then factor 7A will activate factor 10 into factor 10A. A, remember, just means that it's the active form of that clotting protein. And so whenever we've got activated factor 10, that's going to convert prothrombin or thrombinogen into thrombin, and then thrombin will convert fibrinogen into fibrin strands. And fibrin is the product, the end product of the clotting cascade. So also notice that with most of these steps, calcium is required to drive the activation of these various clotting factors. And keep that in mind. We need calcium, and calcium is usually in higher abundance during tissue damage or cellular damage, and that's kind of what helps promote the clotting cascade as well. So just as a review, whenever we've got activated factor 10, or you can also say 10A, just the A means that it's activated, that's going to take prothrombin and convert it into the active form thrombin. And then thrombin will take fibrinogen and activate it into fibrin, and then we've got the formation of these fibrin strands that make up the blood clot and stabilize the platelet plug. So remember, with the extrinsic pathway, we've got tissue, specifically endothelial cell damage and the release of tissue factor, which will activate factor 7. And once we've got activated factor 7, that will promote the formation of active factor 10. And then whenever we've got activated factor 10, then we've got prothrombin converting into thrombin, and then thrombin will convert fibrinogen into fibrin, which makes up those fibrin strands of the clot. On the other hand, we've got the intrinsic pathway over here, where we've got subendothelial collagen exposure, so damage to blood vessels, and when that happens, factor 12 will come across that collagen, and that will activate factor 12. And then factor 12 will activate other clotting factors until finally we're activating factor 10 again. And then the process is the same. So where the extrinsic and intrinsic pathways converge is with the activation of factor 10. So once we have active factor 10, it doesn't matter which pathway activated factor 10. If we've got active factor 10, then we're going to have prothrombin converting into thrombin, and then thrombin will convert fibrinogen into fibrin, which is the main component of those fibrin strands. So here's where I like to ask a couple questions that hopefully get students thinking about this process. So can you have activation of both the intrinsic pathway and the extrinsic pathway at the same time? The answer is yes, you can. And in fact, when you fall off your bike and skin your knee, that's going to activate both extrinsic and intrinsic pathway. So we've got vascular damage, so subendothelial collagen exposure. And if we've got vascular damage like that, then we definitely have damaged endothelial cells along with that. So we have activation of both intrinsic and extrinsic pathway in that example. So can you have activation of just the extrinsic pathway? Hopefully you're thinking about it and not just waiting for me to tell you. But the answer here is yes, you can. And we do see instances where we just have activation of the extrinsic pathway. So remember, what's activating extrinsic pathway? It's damage to endothelial cells. So we've got a number of things that will damage endothelial cells. Tobacco smoking will damage endothelial cells. Chronic hypertension will damage endothelial cells. Sepsis will damage endothelial cells, and so on. So any number of things that will damage endothelial cells or irritate those endothelial cells potentially will activate the extrinsic pathway. So think about a complicated plaque with atherosclerosis. The formation of a fatty streak and an atherosclerotic plaque damages and irritates those endothelial cells. So if those endothelial cells get irritated enough or damaged enough by the presence of that fatty streak in atherosclerosis, then those endothelial cells are going to secrete tissue factor, which will activate factor 7, which will ultimately activate factor 10, which will convert prothrombin into thrombin, and then thrombin will convert fibrinogen into fibrin, and then we've got the formation of a blood clot on top of that atherosclerotic plaque. So then we've got a complicated plaque forming there. And if you remember the complications with that, definitely we'll start occluding arterial blood flow if we've got a complicated plaque forming. But that's just one example of how we can definitely have activation of the extrinsic pathway without activation of the intrinsic pathway. But on the other hand, if we have activation of intrinsic pathway, we definitely will have activation of extrinsic pathway because of the mechanisms involved. Okay, oh gosh, all of these yellow arrows, don't even worry about that. Just keep it simple as what I've been going throughout this section. All right, so let's review. We've got a nail sticking into somebody's foot here, and we have active bleeding. So what do you think is going on here? Is this extrinsic? Is the extrinsic pathway going to get activated, or is the intrinsic pathway going to get activated? And if you're thinking, well, we've got active bleeding going on, so there's vascular damage, then that means that, yeah, we're going to have both intrinsic and extrinsic coagulation cascade pathways activated here. So we've got a torn blood vessel that's resulting in bleeding, and so the first thing that's going to happen initially is vasospasming, but that's only going to stop blood flow for just a few moments, and then we'll have platelet plug formation here. So these platelets get activated, they get sticky with one another, they're going to grab onto each other, grab onto the walls of the blood vessel, trap red blood cells within that platelet plug, and try and form a plug so that blood loss is minimized or stopped. At the same time that platelet plug formation is happening, we have activation of extrinsic and intrinsic clotting cascade pathways. And so both of those will result in the formation of thrombin, and then thrombin will activate fibrinogen into fibrin, and that will create these fibrin strands that will stabilize this platelet plug and stabilize this blood clot. So in this microscopic picture here, these yellow strands are actual fibrin strands. So they're forming kind of a netting or a meshwork that stabilizes this blood clot. And once it's stabilized like this, then platelets can degranulate, they can release their goodie bag contents and start to promote tissue healing and repair. But that's not the last step. Remember, we've got fibrinolysis as the last step as well. So once we form a clot, we want it to go away eventually. We don't want a blood clot persisting because then it might detach and become an emboli. But before fibrinolysis happens, we have clot retraction. So basically, as the tissue is being repaired and heals, the fibrin strands will start retracting, and they shorten, and they will contract that whole clot. And what that will do is help to approximate the wounded edges of the blood vessel, and basically helps it glue itself back together. Remember Virchow's triad? The three characteristics or the three risk factors that almost guarantee a blood clot forming? Remember, we've got changes in the blood vessel wall. That would be like damaged endothelial cells, or even vascular trauma, torn blood vessels. That's going to change the blood vessel wall, and endothelial cells will secrete tissue factor, which will activate clotting cascade. And if there's trauma to the vessel, subendothelial collagen will be exposed, and that will activate the clotting cascade as well. Also, changes in blood flow. Keep in mind that blood is always flowing in a forward direction under normal circumstances. But if blood starts to become stagnant or starts slowing down its flow, then we run the risk of forming a clot because of hypoxic damage to endothelial cells. And then finally, changes in blood composition. This relates to the hypercoagulability of Virchow's triad, of its three characteristics. So if somebody forms too many platelets, then we've got overactivation of platelets and the formation of a platelet plug and blood clot. Or if somebody is being treated with blood thinners and whatnot, and then they don't take their treatments or they're not taking their medicine, then their blood becomes more hypercoagulable, and that will definitely increase the likelihood of forming a clot. So here we've got a full lumen filling clot, a thrombosis for sure. So if we were to take apart this clot, we would find red blood cells for sure. We would definitely find platelets that have been activated, and we would find fibrin strands crisscrossing and stabilizing this entire blood clot. So a situation like this could have been started by endothelial cell damage from chronic hypertension and tobacco smoking or something like that, or even sepsis. And if we get a full lumen clot forming, then we've got an arterial obstruction, and if this is in a coronary artery, for instance, that can lead to a myocardial infarction and possibly even death to that person. Okay, so then finally we need fibrinolysis to occur. We need a way for that blood clot to get broken down and dissolved away. We don't want it staying there forever. So there is another cascade system that's responsible for breaking down blood clots, and we call that the fibrinolytic system. And the main proteins involved here are plasminogen, which is a precursor, inactive form of plasmin. So once plasminogen gets activated into plasmin, it's plasmin that will start breaking down fibrin. It starts piecing it off, starts dissolving fibrin away. And what we're left with are these fibrin degradation products, or these small little pieces of fibrin. And some of those we call D-dimers and whatnot. We can measure fibrin degradation products in the blood. So we can take a blood sample of somebody that we suspect might be forming a lot of clots, and we can verify that if they have a high number of fibrin degradation products. So if there are a lot of FDPs, then that suggests that there's a lot of fibrinolysis going on. And the only way that a lot of fibrinolysis can occur is if there's a lot of fibrin and blood clots occurring as well. So this is just another clinical measurement of what's going on with somebody's blood supply. So here we've got plasminogen, and we've got various things that will activate plasminogen into plasmin. One thing that I wanted to highlight here is that active factor 12 from the intrinsic pathway, not only does it continue the clotting cascade activation, but the presence of factor 12A also will activate plasminogen into plasmin. And so then plasmin will break down fibrin into these fibrin degradation products. So while a clot is forming, it's also being dissolved. So it's not absolute though. When there's tissue damage, endothelial cell damage, or vascular damage, and we've got the clotting cascade activating, forming fibrin clots, it's definitely the factors that are favoring clot formation are stronger than the factors that are favoring fibrinolysis early on. But as the tissue damage starts repairing and healing up, then the factors that are favoring clot formation decrease, and that allows for more of the plasmin to have an influence to dissolve that clot away. So this is a dynamic process. It's not on or off. It just depends on which scenario, which pathway is being stimulated more, the fibrinolytic pathway or the clotting cascade pathways. So let's look at this in a flow chart. So we've got fibrinogen converting into fibrin clot. So all of the clotting cascade steps are packed into this box right here. And once we have fibrin strands forming, we've got the fibrin clot. At the same time, with factor 12A and these other proteins like streptokinase and urokinase and so on, these things will activate plasminogen into plasmin. And plasmin is that protein that will start breaking down the fibrin strands into fibrin degradation products. So if the clotting cascade starts slowing down because tissue is repairing and healing, then that will allow for plasmin to have more of an effect on the fibrin clot, and eventually it will totally be dissolved away, and then we're left with healed tissue It's good as new. All right, lastly, I want to end with just a brief couple minutes explaining that we can look at the blood cells, the elements that are found in the blood, and get a lot of clinical information based on how much or how many of these formed elements or dissolved proteins are in the blood, and so on. So it gives us a lot of really good clinical information. Again, I'm not going to test you on normal values or anything like that. I just wanted to show up a pretty generic looking blood test printout or lab results. So here we've got a complete blood count with differential, meaning that the machine can identify the various blood cells and count those, and then also platelet count. So white blood cell count, we can count how many white blood cells in general there are, and if there's too many or too little, that gives us clinical information as to what's going on with that person. Same thing with red blood cell count. If there's too many or too little or too few red blood cells, then that gives us a lot of clinical information. We can measure hemoglobin and hematocrit numbers. MCV, MCH, and MCHC are morphological measurements of red blood cells, like the volume of the red blood cell, how much hemoglobin's in each red blood cell, and so on. We can count the separate white blood cells that are found in the blood, and so on. So the further along you get in your nursing career and education, you'll learn more how to read these types of things and kind of the clinical information that relates to the lab readouts that we're getting. Okay, so this will end the part 2 of the hematology section. So we've already gone through part 1, obviously, and then part 2 is mostly concerning the hemostatic pathways and the coagulation pathways. Be sure to study up on those, and also that pretty much finishes the normal structure and function of the hematologic system. So the next topic that we're going to talk about are hematologic alterations. So we're going to look at the various types of anemia and what that means, and then looking at some of the bloodborne cancers like the leukemias and lymphomas and so on. So look for that in the future coming up. And as always, if you have any questions regarding what we went over in these videos, be sure to email me those questions and I'll get back to you as soon as I can. All right, thanks guys.